Welcome to Stem Cell Technologies educational webinar. My name is Christy Fable, and I'm the Neural Cell Biology Product Manager here at Stem Cell Technologies. In today's webinar, Dr. Sharon Lewis, Associate Director in Research and Development for Cell Culture Products, will talk about neural stem cell culture systems. Let me now hand you over to Dr. Lewis.
Welcome to our webinar on neural stem cell culture systems. In this webinar, I will describe and compare the Neurosphere and Adherent Monolayer culture systems for the study of neural stem cells from the mammalian central nervous system. In doing so, I hope to provide you with information that will make your decision about which culture system to use easier. I also encourage you to compare the two culture systems in your laboratory and determine which system works best for the experimental questions you are looking to address. The webinar will be divided into the following topics. First, I will provide a brief overview of neural stem cells. I will then describe technical aspects of working with neurospheres in culture, followed by the technical details for culturing neural stem cells under adherent monolayer culture conditions. I will then provide some data directly comparing the Neurosphere and adherent monolayer culture systems using our media NeuroCult. Finally, I will discuss an assay called the NCFC assay, which is useful for quantification of neural stem cells and neural progenitor cell frequency. At the end of the webinar, I will take your questions. Multipotent stem cells, or tissue-specific stem cells, are relatively rare, quiescent cells in the body. There are no unique stem cell markers, and often a combination of markers are needed to enrich for a population of cells containing stem cells. Stem cells are often indistinguishable from other cells in terms of their morphology and phenotype. Therefore, stem cells are often studied in retrospective functional assays designed to show self-renewal ability, multi-lineage differentiation potential, and the ability to repopulate when their numbers have been reduced or damaged. Many sophisticated culture systems have been developed and in vivo systems to demonstrate the long-term repopulation ability of a stem cell, for example, in the hematopoietic, skin, gut, and mammary systems. Neural stem cells can be defined based on a functional criteria in vitro. Neural stem cells are undifferentiated cells. They have the ability to self-renew, produce a large number of progeny or transient amplifying cells over an extended period of time, and maintain multi-lineage potential over an extended period of time. In the central nervous system, there are basically three major mature cell types: neurons, astrocytes, and oligodendrocytes. Neurons are the functional unit of the brain and transmit action potentials through neuronal networks and neurotransmitters. Astrocytes and oligodendrocytes, collectively called glial cells, play a supporting role for the neurons. For example, oligodendrocytes produce myelin sheaths which enwrap the axons of neurons. Astrocytes provide a scaffold for neurons for growth and migration during early development. A population of early neural stem cells begins to emerge at the time of the neural plate and neural tube formation during mammalian embryonic development. These primitive or early neuroepithelial progenitor cells divide symmetrically to increase the pool of stem cells and are responsible for the first wave of neurogenesis. In the developing fetal brain, neural epithelial progenitors give rise to radial glial cells, another population of neuroprogenitor cells which have a more limited differentiation potential than neuroepithelial progenitors. Radial glial cells also act as scaffolds for migrating newborn neurons in the cortex. Finally, in the adult brain, which I will describe later, neuroprogenitors exist in two distinct regions. In these distinct regions of the adult brain, neural stem cells are relatively quiescent and maintain neurogenesis and gliogenesis throughout the adult life. As you can appreciate, there are a heterogeneous population of neural stem cells throughout different stages of embryonic development, but for the purposes of this talk, I will refer to these cells collectively as neural stem cells. Shown here in this slide are the development stages of the central nervous system on the top, and the corresponding in vitro systems and associated publications which describe the isolation of the different neural stem cells in vitro. In this webinar, I will describe some of the in vitro culture systems commonly used to isolate and study neural stem cells from the fetal and adult brain, and referring to the publications below. First, it has been shown that neural stem cells can be located in many regions of the fetal or embryonic mammalian brain. For example, tissues are microdissected from the ventral mesencephalon in the ventral region of the embryonic day 14 mouse brain, or from the cortex and striata of the E14 mouse forebrain. Most studies employ fetal tissues taken from mouse or rat embryos ranging from embryonic day 10.5 to 18 days. The presence of neural stem cells has been extensively investigated in two regions of the adult mammalian brain, the subventricular zone, SVZ, and the hippocampus. Shown here is the SVZ region, which is a thin layer of cells that lines the ventricles of the lateral ventricles in the forebrain. The SVZ region contains cells which have self-renewal ability and are the cells which give rise to new neurons in the olfactory bulb throughout the life of the mouse. Neural stem cells produce neuroprogenitors, which migrate the rostral migratory stream and differentiate into interneurons within the olfactory bulb. The SVZ region is obtained by microdissection techniques. The second region in the adult brain containing stem cells is the hippocampus. The hippocampal progenitor cells are also obtained by dissecting the dentate gyrus within the hippocampus. The progenitor cells from this region respond to EGF and FGF. To induce differentiation, FGF is added to culture to promote differentiation into glial and neurons, while BDNF promotes neuronal survival and differentiation in vivo. The progenitor cells within the hippocampus are believed to maintain hippocampal function with ongoing neurogenesis, which is important for memory and learning. As I mentioned previously, neural stem cells can be obtained from all stages of the developing embryo, from the most primitive developing neuroectoderm, the fetal brain, until the adult brain stage. Neuroprogenitors, which include stem cells, can then be studied using the Neurosphere culture or the Adherent Monolayer system. While the Neurosphere culture system has been most widely and extensively used for over 15 years, it's only recently that the culture conditions for the Adherent Monolayer system has been optimized to produce long-term adherent cultures from mouse and human cells. In both systems, a population of cells which have the ability to self-renew and demonstrate multi-lineage potential has been isolated. I will first describe the Neurosphere culture system. While several early research indicated that there was some proliferation of cells in the adult brain, most of the work reported limited proliferation of these cells and that these cells were species-specific. It was not until the early 1990s that Reynolds and Weiss were able to isolate and culture a population of cells from the adult mouse brain that showed the properties of a stem cell. They developed the culture system, called the Neurosphere culture system, in which they could show that cells isolated from the adult brain were undifferentiated, had the ability to self-renew, was able to produce large numbers of progeny, and maintained multi-lineage differentiation over an extended period of time. Therefore, using the Neurosphere culture system, Reynolds and Weiss were able to demonstrate that the population of cells which exhibited neural stem cell properties existed. By definition, a cell that meets all of the criteria listed here is a stem cell, and a cell that meets only one or some of the functional criteria is not a stem cell, but is defined as a progenitor cell. The Neurosphere culture method that Reynolds and Weiss developed is shown here. In brief, tissue is microdissected from different regions of the embryonic or adult mammalian brain and processed into a single cell suspension. The cells are then plated in serum-free media containing the mitogen EGF or a combination of EGF and FGF, either in bulk cultures or at low density in wells. After about 5 to 7 days, depending on the species tested, a rare population of cells will respond to EGF or EGF and FGF, depending on the culture condition, to form small clusters of cells. These will eventually float in suspension. These clusters, termed neurospheres, continue to grow in diameter. At some point, these neurospheres have to be dissociated in order to avoid the center cells going necrotic and dying. The neurospheres are dissociated into a single cell suspension using various methods, and the single cells are replated under the same conditions as the original primary cell. The entire procedure can be repeated over and over again to generate new neurospheres. As long as there are stem cells which self-renew and produce more progeny, i.e. stem cells, new neurospheres can continue to be generated. Over time, if you count the total number of cells and graph that to the number of days in culture or passage number, a linear curve can be generated showing an expansion in the total cell numbers over time from the starting sample. In theory, the only way you can see an increase in total cell numbers over time is if stem cells are maintained and they self-renew and produce a large number of progeny. If cells were to be removed at different times during the expansion phase of the culture and exposed to serum and adhesion substrate, the cells would immediately differentiate. If neural stem cells are maintained throughout the long-term culture, then it would be possible to detect the three main cell types of the CNS, which are the neurons, astrocytes, and oligodendrocytes, as shown here. In red, the neurons have been stained with an antibody against beta-tubulin. In blue are the astrocytes stained with an antibody against GFAP, and in green, oligodendrocytes are stained for an antibody against the marker O4. Therefore, by applying the Neurosphere culture system in this manner, i.e. long-term cultures and differentiation at different time points, you can demonstrate the presence of neural stem cells. We have basically used the original media formulations and protocols and developed an entire product line called NeuroCult. NeuroCult provides the necessary media and reagents required to work with neurospheres and neural stem cells. I will describe the tools provided for the expansion, the differentiation of neural stem cells, the dissociation of neurospheres, as well as functional assays to characterize neural stem and progenitor cells. The key features of our product line is that our media formulations and protocols have been developed based on the original Reynolds and Weiss publication. The media formulations are species-specific, and they have been designed to isolate and expand neural stem and progenitor cells. High-quality media reagents are used, and the components are pre-tested and selected to support the desired application to minimize variability. Each batch is performance tested for consistency and reproducibility in the Neurosphere cultures, and no surrogate assays are used. By doing these things, we hope to allow for standardization within and between labs. The Neurosphere culture system has been a valuable system for the large-scale expansion of neural precursor cells. Here I will describe some of the major technical details that need to be observed when working with neurospheres. Once tissue is dissected from the embryonic or adult brain, the tissue has to be dissociated using mechanical and/or enzymatic means into a single cell suspension. Shown in the slide are the media and protocols we have developed to obtain consistent and robust neurosphere cultures. For mouse cells, cells are counted and seeded at a specific cell density in NeuroCult complete media containing EGF or EGF and FGF for adult mouse cells. After 5 to 7 days, large numbers of neurospheres are generated in response to the growth factors. The neurospheres are dissociated when they reach about 100 to 150 microns in diameter to produce a single cell suspension. and these are counted again and replated under the same culture conditions as the primary cell. The cells can be either plated in bulk to generate large numbers of cells or plated clonally in wells to produce clonal neurospheres depending on the applications. At any time during the culture's intact neurospheres or the single cells from these neurospheres can be exposed to neurocult differentiation supplements in order to obtain differentiation into the three cell types, the neurons, astrocytes, and oligodendrocytes. Using neurocult complete media, you can consistently achieve reproducible long-term cultures of neurospheres derived from either the embryonic or the adult mouse CNS tissues. There is high expansion of total cells over time, indicating that the media and protocols are maintaining neural stem cell populations very efficiently. High cell expansion in long-term cultures beyond passage 5 is an important criteria to demonstrate self-renewal ability and compare to other media formulations. Neurocult is an ideal media for long-term cultures for mouse CNS cells. Another indicator of a healthy neurosphere culture is the actual morphology of the neurospheres in the culture medium. At day 1, small clumps of cells are normally attached to the bottom of the tissue culture vessel, and the cells continue to proliferate within these clumps. As the clumps or neurospheres increase in size and density, they detach from the bottom of the dish and float in suspension. By day 6, the majority of neurospheres should be floating in suspension and be transparent spheres with round, intact peripheries. The cells within the neurospheres are phase contrast bright without any dark necrotic centers. At high magnification, you should be able to see small microspikes at the edges of the cells, and these are thin protrusions which act as feelers of the microenvironment in the media. Moving on to rat CNS cells. Some of the common problems that researchers experience with neurosphere cultures derived from rat CNS cells is the stickiness of the cells. The growth requirements for rat cells are specific and slightly different from those of mouse CNS cells. Because rat cells tend to stick to the bottom of the flask and differentiate, as shown here with the red arrows, one of the common problems is the lack of success in obtaining long-term neurosphere cultures from rat cells beyond passage 5. We have therefore developed a rat-specific media formulation and the protocols to support neural stem cell cultures from rat CNS cells. The complete media formulation for rat has been optimized to produce long-term cultures when used in combination with EGF, FGF, and heparin. The protocols for neurosphere cultures for rat cells have also been optimized. For example, the culture period for rat cells is 4 days compared to mouse cells, which is 5 to 7 days. A half media change or media replenishment step is included 2 days after cells are plated. At day 4, the neurospheres derived from rat cells are dissociated into a single cell suspension, and these can be replated under the same culture conditions as the first culture. At any time, the cells can be induced to differentiate into neurons, astrocytes, oligodendrocytes in the complete differentiation medium. We compared E14 rat cortical cells cultured in our media to a traditional media formulation, either in T25 flask or in ultra-low adherent culture dishes as recommended by the supplier. We found that the overall expansion of rat cortical cells was higher in our media, shown in green, compared to the traditional media formulation, shown in gray, in the T25 flask. The use of ultra-low adherent culture dishes to prevent cell sticking was not necessary, and shown here, cells cultured in our media, in green, or the traditional media formulations, showed an overall lower total cell expansion. Therefore, the optimal conditions for generating long-term cultures from rat CNS cells is to use neurocult media in T25 flasks. We have also developed a specific media formulation for the culture of cells from the human CNS. Shown here are fetal cells from 2 different regions of the human brain, cultured in our media for over 40 days, showing the standard growth curve for high total cell expansion over time. The neurospheres generated in those cultures are shown here. The neurospheres derived from human CNS cells look similar to those derived from mouse and rat cells, and it's important to maintain these neurospheres within the size of 100 to 150 microns in diameter. As you know, one of the steps routinely used in the neurosphere culture protocol is the dissociation of the cells in order to generate a single cell suspension. It is important that the starting cell population for a neurosphere culture is a good suspension of single cells with high viability. We have developed various methods to produce viable single cell suspensions from neurospheres. A common problem with the neurosphere culture is that the decision as to when the neurospheres are ready to be dissociated and the cells ready for passage into the next culture. One way to estimate when to passage the neurospheres is to take an aliquot of neurospheres from the culture and dispense this aliquot directly through a hemocytometer, shown here. Using the grid lines as a guide, a 100 micron diameter neurosphere should fit within 2 grid lines as shown by the red lines. If the neurospheres are between 100 to 150 microns, they can be passaged. Leaving the neurospheres to grow larger than this will cause cells in the center to go necrotic due to decreased exchange of gas and nutrients. With experience, it will also become evident to the researcher when the neurospheres are overgrown or ready for passaging. Leaving the neurospheres to grow large will result in the edges of the spheres to start breaking off, and they will no longer be smooth. Compare a poor neurosphere culture to a good neurosphere culture with intact and round peripheries. The cells within the neurospheres are usually translucent and bright and should not be dark. The neurospheres shown in F are ready to be dissociated and the cells passaged. We have developed a chemical dissociation kit which provides a non-enzymatic and non-mechanical method to dissociate neurospheres. A common method of breaking up neurospheres is to use the mechanical procedure called titration with the use of plastic or glass pipettes. This mechanical procedure is very harsh and often results in high cell death. The chemical dissociation solutions are designed to release the cells from the extracellular matrix within the neurospheres. The neurospheres are harvested by centrifugation and the supernatant completely removed. Solution A is added directly to the pellet containing neurospheres and the spheres are suspended. Solution B is then added and immediately a timer is set for 8 minutes, during which time the cells are mixed gently at 3 and 7 minutes. No titration is required. At the end of 8 minutes, solution C is added and the mixture is suspended gently. Finally, neurocult media is added and a single cell suspension is generated and ready for density and viability counts. We show here the total cell expansion for cells which are treated at each passage with the chemical dissociation solutions in green or mechanical titration shown in blue. As you can see, the expansion of total cells is higher for the cells treated with the chemical dissociation kit compared to the titration method. And this may be due to the increased cell viability at each passage that was higher for the chemical procedure. The cells treated with this chemical dissociation kit continue to maintain all functional properties of a stem cell. We have also used acutase enzymes to dissociate neurospheres. Here, the neurospheres were dissociated at each passage with either acutase, shown in green, or the titration method in gray. The cell viability was higher when neurospheres were dissociated with acutase, shown here in green, and this resulted in an overall higher expansion of total cells in long-term neurosphere cultures. We have now also developed this differentiation media for mouse, rat, and human CNS cells. When intact neurospheres or the single-cell suspensions obtained from the dissociated neurospheres are exposed to low levels of serum and a matrix, the stem cells and progenitor cells differentiate immediately into neurons, astrocytes, and oligodendrocytes. The mature cells are routinely detected with antibodies against lineage-specific markers. Here, the neurons were detected with an antibody against MAP2 or beta-tubulin. Astrocytes were detected with an antibody against GFAP, and oligodendrocytes were detected with an antibody against MBP. Immunocytochemistry antibodies are available also, and these have been pre-tested. I have so far talked extensively about the technical aspects of working with the Neurosphere culture system. I will now move on to describe the technical aspects of working with neural stem cells and progenitor cells using the Adherent Monolayer culture system. I will also present some data where we have compared cells cultured in the Neurosphere culture system versus the adherent system. In these studies, we have used the same media formulation, i.e. NeuroCult, to compare the two systems, since the media plays a significant role in the functional properties of the cells, and this also in order to avoid variability in the results due to differences in media formulations. So the main features of the adherent culture is that cells are cultured in a serum-free media which must contain EGF and FGF in the presence of a substrate or extracellular matrix. In earlier studies, the lack of success in obtaining consistent long-term adherent cultures from mouse and human CNS was due to the use of only EGF or FGF alone. It was also shown that certain substrates like laminin are better at supporting long-term cultures compared to poly-D-ornithine or fibronectin. Basically, the presence of a substrate promotes cell adhesion to the substrate and prevents cell-to-cell aggregation, or in other words, neurosphere formation. Some publications have reported that cells cultured in adherent conditions are niche independent and therefore promotes a more homogeneous populations of cells in the culture. And this was based on the expression of nestin, RC2, PAX6, and CD133, for example. This population is believed to be the radial glial cells present during embryonic development. The Neurosphere culture, on the other hand, is believed to produce more of a microenvironment mimicking the brain niche, and as such promotes differentiation within the neurospheres and a more heterogeneous population. It is also claimed that neural stem cells are better maintained in the adherent cultures due to more symmetrical divisions, while asymmetrical divisions occurs in the neurospheres. However, these results were based on the number of cells expressing nestin and other markers for radial glial cells, and the lack of expression of markers for differentiated cells within the adherent cultures. Marker expression does not provide complete information on the functional properties of the cells, and nestin is not a good marker for stem cells. And in none of these studies were neural stem cell numbers measured directly. The adherent cultures are also reported to maintain neurogenic potential for the cells compared to the neurosphere cultures, which tend to produce more glial progenitors. The adherent culture system has also been shown to isolate tumor-initiating cells or tumor stem cells. The technical features of the Neurosphere and adherent culture systems are outlined in this slide. The Neurosphere culture system does not require a substrate. Adherent cultures require the plates and wells to be pre-coated with PLO or laminin prior to initiating the cultures. This requires some preparation time, and the coated plates are not stable for long periods of time and should be used immediately. Cells form aggregates in suspension in the Neurosphere cultures, while cells attach to the substrate and proliferate in the adherent system. The success of obtaining a confluent culture is variable and can depend on the media and cytokines. Neurospheres are dissociated by various mechanical, enzymatic, and non-enzymatic procedures. Adherent cultures have to be detached by enzymes, and sometimes the effects of enzymes on cell surface protein and cell functions needs to be investigated. Long-term cultures can be generated from E14 mouse cells in EGF containing media only for the Neurosphere cultures. For adherent cultures, EGF and FGF have to be used. The adherent culture system therefore offers an alternate culture system to isolate and culture neural progenitor cells. In some cases, different media was tested for the Neurosphere versus the adherent culture system as in publications, and this may account for some of the differences in the results. It is therefore important for side-by-side comparison of the two systems to be performed using the same media conditions and cytokine combinations. We have now developed protocols for adherent monolayer cultures using our NeuroCult reagents and media. We tested NeuroCult in adherent monolayer cultures for both embryonic day 18 and adult mouse cells. Tissue was dissected and processed according to the procedures outlined earlier in my talk. The single-cell suspension was then seeded at the specific cell density on laminin-coated flasks to generate adherent cultures. The cytokines EGF, FGF, and heparin cofactor was included in the media. The cells were detached at each passage with acutase. The resulting cells from the adherent cultures were analyzed for their functional properties. When culturing primary SV cells in adherent culture systems, we noticed that primary cells, when first seeded, contain a lot of debris from the original tissue. However, by 7 days, a monolayer of adherent cells can be observed. In subsequent passages, less debris is observed as it is washed off, and the adherent cells can be seen by day 3. After 5 to 7 days, the cells would proliferate until they reach 70 to 80% confluence, which is the time the cells should be harvested and replated. Cultures at day 7 can be immunostained for markers of undifferentiated cells, and shown here are cells cultured in NeuroCult in the presence of EGF and FGF that express high levels of nestin. We have compared the use of various substrates for the expansion of cells in adherent monolayer cultures. We found that laminin alone showed a consistent high total cell expansion of adult SPZ cells compared to a combination of laminin and PDL or laminin and polydornithine. Therefore, laminin can be used with our media for the expansion of neural stem cells and progenitor cells from mouse CNS cells. We also tested the growth of SVZ adult cells in adherent monolayer cultures using the NeuroCult formulation or different media formulations used in the literature. We found that NeuroCult efficiently expanded neuroprecursor cells at high levels compared to the other formulations tested, including the routinely used N2B27. Interestingly, we found that NeuroCult was highly effective at supporting the growth of primary or freshly dissected adult brain tissues compared to the other media, indicating that our media was supporting total cell expansion and possibly not selecting a select population to survive in the early cultures. We tested NeuroCult media in Neurosphere and adherent monolayer cultures for both embryonic day 14 and the adult mouse CNS cells. We compared the two cultures side by side. So single cells were seeded at specific densities in either uncoated flask or laminin-coated flask to generate adherent cultures. EGF, FGF, and heparin cofactor was included in the media, and in both cases acutase was used to either dissociate the neurospheres or to detach the cells from the monolayer, from the flask. The resulting cells from the neurosphere and the adherent cultures were then analyzed for their functional properties, including self-renewal, differentiation, and stem cell numbers. As you can see here, the total expansion of cells is higher in the adherent monolayer conditions compared to the Neurosphere culture system. The mean fold expansion per passage was 16-fold in the Neurosphere cultures compared to 25-fold in the adherent cultures. Therefore, total cell expansion is higher in the monolayer condition. However, later in my talk, I will provide data on whether the total expansion of cells was also correlated with an increase in neural stem cell numbers. Looking at differentiation, at passage 1 and passage 5, Cells from dissociated neurospheres in gray and adherent monolayer cultures were differentiated in the presence of differentiation supplements. We counted the number of neurons, astrocytes, and oligodendrocytes. The results showed that about 30% of the differentiated cells were neurons in both the Neurosphere and adherent cultures at passage 1. And this percent dropped to 18% for cells derived from the Neurosphere cultures compared to 10% for the differentiated cell population derived from adherent monolayers. Oligodendrocytes were maintained at 0.1% of the differentiated cell population derived from either the Neurosphere or the adherent cultures at passage 1 and passage 5. The percent of astrocytes was approximately 40% in cells derived from the adherent cultures compared to 25% for differentiated cells from the Neurosphere cultures. By passage 5, both adherent and Neurosphere cultures generated 70 to 80% astrocytes when differentiated. Therefore, the results indicated that cells cultured in the Neurosphere or adherent monolayer cultures retain the ability to produce the 3 cell types of the CNS, and the proportions of each mature cell type is similar. I have described the basic technical features for working with neural cells in the Neurosphere and the adherent monolayer cultures to study the functional properties of neural stem cells and neural progenitor cells. Now I will talk about another assay that you can use to measure neural stem cell numbers and neural progenitor cell numbers. The neural colony forming cell, or NCFC assay, is a single-step assay to enumerate neural stem cells, neural progenitor cells. Both the Neurosphere and the adherent monolayer culture system does not allow the quantification of neural stem cells and neural progenitor cells directly. Going back to our definition of a neural stem cell, a cell that is undifferentiated has the ability to self-renew, produce a large number of progeny over an extended period of time, and maintain multilineage differentiation over an extended period of time. A cell that fulfills all of the criteria listed is defined as a stem cell, and a cell that only meets one or some of the criteria listed is termed a progenitor cell. One of the major limitations of the Neurosphere assay is that not all cells capable of producing a neurosphere fulfill all of the criteria of a neural stem cell. In other words, a progenitor cell can form neurospheres as efficiently as a neural stem cell. Therefore, how do you know if a neurosphere was generated by a stem cell or a progenitor cell? Only a subset of neurosphere-forming cells are therefore stem cells. Sphere number is not an accurate measure of neural stem cell frequency and in fact may overestimate the neural stem cell numbers. It is for that reason we developed and published the application of the NCFC assay as a more accurate assay to measure neural stem cells and neural progenitor cell numbers. The NCFC assay is outlined here. You can start with dissected mouse CNS cells or cells generated from Neurosphere cultures. A single cell suspension is obtained and it's filtered and then counted. A specific number of cells are then mixed with NCFC media, cytokines, and cytokines. Collagen semi-solid media is then mixed in and the entire suspension is plated in 35mm dishes in duplicates. The cells are allowed to grow for 21 days, during which time clonally derived colonies will be seen. After 21 days, colonies of different sizes can be observed. The size of the colonies is correlated with the proliferation ability of the original colony-forming cell. Therefore, a cell which has high proliferative potential will form very large colonies, while cells with more limited proliferation potential will form smaller sized colonies. We characterize all the colonies in the NCFC assay based on their diameter into 4 categories. Colonies greater than 2 millimeters in diameter, colonies between 1 to 2 millimeters, colonies between 0.5 to 1 millimeter, and finally very small colonies which were less than 0.5 millimeters in diameter. We found in many experiments that the colonies generated in the NCFC assay fitted well within these colony size categories as shown here. We performed extensive validation experiments, and this is published in detail in our paper. We proved that cells isolated from the colonies greater than 2 millimeters in diameter were able to generate long-term neurosphere cultures, and there was an expansion in total cell numbers over time. The cells from colonies less than 2 millimeters in diameter could not generate long-term neurosphere cultures. Therefore, the cells which generated the colonies greater than 2 millimeters in diameter fulfilled the functional criteria of self-renewal and production of a large number of progeny. The cells from the colonies greater than 2 millimeters in diameter which generated neurosphere cultures were also assayed for their ability to generate neurons, astrocytes, and oligodendrocytes. We found that cells from colonies greater than 2 millimeters were able to produce the three main cell types immediately or after they were isolated from the colonies and then passage multiple times in the neurosphere culture. The cells from the smaller colonies only produce 2 or 1 mature cell type. Therefore, only colonies greater than 2 millimeters in diameter contain cells which self-renewed, produced a large number of progeny, and shown here maintain the ability to produce neurons, astrocytes, and oligodendrocytes, therefore fulfilling all the criteria of a neural stem cells. The colonies smaller than 2 millimeters in diameter were produced by progenitor cells. Therefore, the NCFC assay is an assay to discriminate neural stem cells from progenitor cells. We have validated that the cells from colonies greater than 2 millimeters in diameter meet all the functional criteria for a neural stem cell, and the cells from colonies smaller than 2 millimeters in diameter are derived from progenitors. The NCFC assay is a useful assay to enumerate neural stem cells and neural progenitor cells, and therefore we used it to compare the number of neural stem cells and neural progenitor cells from Neurosphere cultures and adherent monolayer cultures. I showed you previously that the expansion of total cells in the adherent monolayer culture system is higher compared to the Neurosphere culture system. Do we see an expansion in neural stem cells? Using the NCFC assay to measure neural stem cells numbers at different passages in the Neurosphere or the adherent monolayer cultures, we found that the high expansion of total cells in the adherent cultures is mainly due to the increase in progenitor cell expansion as seen in the large number of colonies smaller than 2 millimeters in diameter. However, as you can see, the number of neural stem cells as measured by the number of colonies greater than 2 millimeters in diameter is comparable for the Neurosphere culture and the adherent monolayer cultures. In summary, I have described different culture systems which are useful for studying neural stem cells. Each culture or assay system has specific applications and properties. For example, the Neurosphere culture system is an excellent system for the isolation and expansion of neuroprogenitor cells from the mammalian CNS cells. You can passage cells long-term, and by doing so obtain large numbers of neuroprogenitor cells. The neurospheres can be used to measure the number of sphere-forming cells or proliferating cells if you're careful to plate at low cell density to avoid aggregation. The Neurosphere system is not a clonal system unless you plate one cell per well and generate clonally derived neurospheres this way. The NCFC assay, on the other hand, is a quantitative assay to measure neural stem cells and neural progenitor cells. Because this system is clonal, colonies are derived from a single cell. There is no aggregation or fusion, so that colony formation is indicative of the properties of the original colony-forming cell. Now comparing the applications of the Neurosphere versus the adherent culture system. Both systems allow the isolation and expansion of large numbers of neural precursor cells or progenitors. In the adherent system, a coating is applied to the tissue culture to promote adherence. Therefore, pre-coating is required before the start of an experiment, and the variability in the coating and substrate quality must be considered, and that can affect the success of the cultures. The neurospheres can be dissociated using many different methods, and the use of enzymes is not always necessary. For adherent cultures, enzymes are the most commonly used method to detach the monolayer of cells. In some cases, the use of enzymes has been shown to cleave surface proteins, which in turn could affect the functional properties of cells, specifically the stem cells. The expansion of cells in the adherent culture is higher compared to the Neurosphere culture system. However, we have demonstrated that the expansion is mainly due to an increase in the progenitor populations. The Neurosphere and Adherent Monolayer culture system both support the population of cells that have the ability to self-renew and demonstrate multi-lineage potential, indicating that neural stem cells can be isolated in both systems. The Neurosphere culture system allows you to study neural stem or progenitor cells within a niche-like microenvironment, mimicking the microenvironment of the mammalian brain, while the adherent monolayer system allows you to study these cells in a niche-independent way. Both systems allow you to isolate tumor-initiating cells or tumor stem cells. Finally, all the protocols used to study neural stem cells in vitro will help us understand the tremendous potential these cells may offer in terms of the clinical applications to treat neurodegenerative diseases and injury. Some of the main areas of clinical research include the treatment of Parkinson's disease, demyelinating disorders, and spinal cord injury, to name a few. It is anticipated that in vitro culture systems will one day facilitate the transition of basic research to actual clinical applications, and some of this type of work has already begun. I would finally like to thank my colleagues at Stem Cell Technology and also all of our collaborators